Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the RNA processing in eukaryotes. Okay, and in my previous video I have already explained about the transcription process, so you check out that video from my channel. Okay, so this is the RNA processing in the eukaryotes. The RNA processing that involves three processes. One is the 5' capping, 2 is the 3' cleavage or polyadenylation, and 3 is the RNA editing. Okay, the three important processes are involved in the RNA processing procedure. Okay, so this is the 5' capping. In eukaryotic, mRNA has an enzymatically appended cap-like structure. Okay, in the eukaryotic mRNA, that has a cap-like structure and which is consisting of 7-methylguanosine residue. Okay, what is there? That means in case of eukaryotic mRNA, there is a cap structure which is consisting of 7-methylguanosine residue, and that is joined via a 5' triphosphate bridge. Okay, so this 7-methylguanosine residue, this joined with each other, that by the 5' triphosphate bridge. Okay, during the transcription, this 7-methylguanosine is added to the 5' end of the nascent mRNA. So what is happening during transcription process? This guanosine that is added with the mRNA 5' end. Okay, and the first step is that RNA capping are catalyzed by a dimeric capping enzyme. This capping process is catalyzed by one enzyme which is dimeric in nature, and that is associated with the phosphorylated carboxyl terminal tall domain of RNA polymerase II. So this dimeric enzyme that is associated with what? This is associated with the phosphorylated carboxyl terminal tall domain, or in the short form you can say CTD, and it is present in what? In where? Not what? In where? In the RNA polymerase II. Okay, and one subunit of the capping enzyme, the subunit means the enzymes has subunits, different subunits are there in the enzyme, and the one subunit among them that removes the gamma phosphate from the 5' end of the nascent RNA emerging from the surface of an RNA polymerase II. So what is the role of one subunit? That one subunit that helps to remove this gamma phosphate substance, that is from where? From the 5' end of the RNA. And the other subunit, the role of other subunit is that it helps to transfer the GMP moiety, okay, guanosine monophosphate moiety, from GTP to the 5' diphosphate of the nascent transcript, or you can say the mRNA. So this is the function of the one subunit, and this is the function of the other subunit. The other subunit that helps to transfer the GMP from where? From the GTP, that is from GTP to where? To the 5' diphosphate of the RNA. Okay, and in that way it try to create what? That guanosine 5' triphosphate structure. In that way this subunit creates 5', obviously guanosine 5' triphosphate structure. Okay, and in the final step what happened? Another enzymes are there that transfers the methyl groups. Okay, that transfers the methyl groups from where? From the 5' adenylyl methionine to where? To the N7. This is 7, okay? N7. N7 position of the guanine at where? At the 5' end of the RNA. So another enzyme, what is the role of the another enzyme? The another enzyme's role is that the methyl group is transferred from the 5' adenylyl methionine to the N7 position of the guanine at the 5' end of the RNA. Okay, this is the role of another enzyme. Obviously, this is the separate enzyme. Okay, so in that way, the capping process, or you can say the 5' capping process, is completed. Okay, and here important thing is written that which is called the cap 0, which is called the cap 1, and which is called the cap 2. The cap 0, which is called if mRNA has a methyl group on the N7 position of the guanine at the 5' end, then it is called the cap 0. Clear? So where the methyl group must be present? Must be present in the N7 position of guanine at the 5' end. Clear? And this is the first methylation step. Obviously, this is the first step, and that occurs in all the eukaryotes. Clear? This cap 0 formation is seen most of the eukaryotes, in most of the eukaryotes. But in some species, a methyl group is added to the second as well as third nucleoside of the capped mRNA. It may be possible in some cases that the second position and the second position or the third position of the nucleoside, these are involved in this capping process. And here the methyl group on the N7 position of guanine and the 2' hydroxyl position of the second nucleotide. Okay, so what is saying that mRNA, here the mRNA, this has the methyl group where in the N7 position, okay, of guanine, and the 2' hydroxyl position of the second nucleotide. Clear? So at the 5' end is known as the cap 1. So what is saying that? That the mRNA with the methyl groups on the N7 position of guanine and the 2' hydroxyl position of the second nucleotide at the 5' end. In that situation, the cap is called the cap 1. Okay, and if methyl group is present at the both second and third nuclear site, so then it is called the cap 2. Okay, clear? Achha, okay. So now we have to understand that functions of the 5' cap. What actually the role of the 5' cap? First one, it gives the protection mRNA from the degradation. Second, transport the mRNA from nucleus to cytoplasm, and binding of ribosome with the mRNA. These the three functions are there that are controlled by the 5' cap. Okay, so